Pereira Santos, and this work is entitled An Efficient Model for Identification of Hidden Peptides with Biological Activities. This work was advised by Professor Mariana Quezada and Professor Sergio Costa from Federal University of Minas Gerais, here in Brazil. These are our collaborators, and this work was done in Laboratório de Biofísica de Macromoléculas. Data science is the technology most important in the 21st century, as we can give good use to our data. As scientists, we have a lot of experimental data that can be used with data science to make better decisions, actions based on trends, test our decisions, and identify new opportunities. What is my goal? My goal is to identify new cryptides. And you can ask me, Bruno, what are cryptides? Cryptides are peptides that comes from larger proteins and can have functions that can be correlated to this precursor protein, or non-related activity, or an activity only identified in vitro. How we can do that? We can do that calculating physical chemical properties that can be done using the peptide sequences. As shown in this work from Spoon and collaborators in 2019, the physical chemical properties are directly correlated with the antibacterial activity of these peptides, for example. So our goal was to develop a new tool capable of simulate a protein degradation that would be able then to create a whole database full of physical chemical features and use machine learning tools to extract from deposited databases information to predict our peptides with biological functions. Also use an evolutionary algorithm to propose peptides improvements. How we do that? We do that using a simple FASTA file that if they are proteins, we need to make this simulation of proteolysis using molecular weight information and enzymatic degradation simulation. And then we generate a lot of peptides, or we can use FASTA files full with peptides, then we jump the proteolysis step, and then we calculate the physical chemical features. As I said, we use the deposited databases to make a supervised classification of these peptides and predict the activity of these peptides. As a case of study, we have this protein from Schistosoma mansoni that our group studied, and we predicted antimicrobial peptides from this protein, and we generated a model with 87% of accuracy. We tested 3 algorithms and we chose random forest to do it. And we also wanted to make improvement in these peptides, because we could probably have better peptides related to function. How we do that? In steps of crossover and mutation that takes the sequence and modifies it to generate improved peptides. We did a step of proof of concept where we chose 3 peptides from the machine learning study and 1 peptide from the genetic algorithm study to synthesize and test antibacterial assays and antifungal assays. As we can see here, 2 of these peptides show high MIC, but 2 of them showed low MIC, and 1 of them was the improved peptide that showed greater antibacterial antifungal activity. We can also use this tool to extract new informations and the unsupervised classification. And in conclusion, we were able to create a tool capable of identifying peptides with biological activity that will help in making good decisions for in vitro studies. I want to thank my collaborators and the funding agencies.